have 5 lights on, and it's lights out, and away we go. Move the crowd when the beat goes down. I rock the stage, they all know my sound.
GM everyone, and thanks so much for joining. Welcome to season 2 of Fly Racing. With us, we have a ton of people, scientists, friends, and all the people who are help executing the fly races. And so, kind of just as a reminder for those of you who are tuning in to Pump Science for the first time, we are a longevity project built on top of Solana, and we are on a mission to find the compounds that help us live forever. And so, yeah, with that, would love to do maybe a quick round of intros, and then we can jump right in. Jillian, go ahead.
Yeah, hey, everybody. I'm super excited for this fly stream. This has been like weeks in the making. So excited to, unleash the flies today. Jillian, co-founder of Pump Science. Quick background on me. I spent some time in big pharma early in my career, doing marketing for new drug launches, and fell down the DSci rabbit hole. And am excited to be building Pump Science to basically, really create open access to research funding, where really all you need is an idea. And then, we have the labs to help you run it. Let's kick it over to Paris. You want to go next?
Hello. Yes, I'm Alberto Parra, a scientist, a PhD, and I'm doing the mouse experiment of the longevity with my CRO. I'm very happy to be part of this project because it's not that it's only interesting, it's like I'm having fun with this.
Oh, having so much fun that you're trying to run away, hence why you're in the car.
Yeah, sorry, here in Europe, it's late, I have a dinner, and I have to do the streaming from here.
Awesome. Definitely taking part in the races yourself. Sam, wanna go next?
Absolutely. It's my name is Sam Banerjee. I'm the co-founder and CEO of Julian Health Sciences. I'm super excited to be here and what this Pump Science platform is doing and providing solution, which is we're all been pursuing for so long. And I think this is a great people, great platform. And I'm super excited to be a part of this fantastic mission. Good to see you guys again.
Back at you, Sam. Victor, want to go next?
So I'm Victor. I'm CTO at Juvion, and I'm very excited to have this live now. We've been working on this for a couple of months now, so it's very exciting.
Yeah, so maybe to give everyone a bit of context, Sam and Victor work at Juvion Health Sciences, who have been running all of the fly races that we're now about to show you. So huge shout out to them. It's been a Herculean effort to get it here, but we finally have it up and ready for you to look at and for us to kind of dig into. Girish, want to go next?
Yeah, absolutely. No, great to be here. I'm a scientific advisor for Rapamycin Longevity Labs, and I work for Ageless RX as a research manager there. It's my full-time job, and just really excited to see how we can fly from these fly experiments to human translational studies. There's some really exciting compounds in here that have a lot of potential. So super excited that we have this accelerator now in the system with Pump Science.
Hell yeah. Krister, you're practically a DSci celebrity at this point.
Yeah, Krister here. I have a IT and business development background, and I'm also the founder of the Rapamycin Longevity Lab, where we take the mTOR pathway to the next level, and this we're going to do by developing different combinational therapies on mTOR inhibitors. So yeah, super exciting to be here, and Pump Science is pushing things forward in a new way. So I fully support that.
Let's go. Awesome. John, would you mind letting my stream into the stream?
You got it.
And then we can just jump right into the races. So for those of you that haven't been onto the Pump Science platform, the way you get to any of the different fly races is you make sure you're on the live tab. So any live experiment you can access from here. You click on any compound. So you'll see right now we have 12 compounds that are being tested. You just click on one. And then you go to the racing tab. So the racing tab's where all of the races will go. And yeah, so here you'll see the average of all the flies that are receiving the compound will be on the left. The average of all the flies receiving the control will be on the right. We test with 6 tubes of flies. So the 3 here, each one has 15 flies in it to start. Of course, the flies die over time. And on the right, these 3 flies receive control. And so basically no drug. And then the tube of flies gets dropped. So all the flies go to the bottom, and then we race them to the top. I'll show you what that looks like in a second, but just want to make sure we have everything covered. And then the flies kind of run around their tube. We measure their speed and their distance traveled. And so what you get at the end of it is a chart where you can see, okay, the BXB, this drug here, is making the flies move faster with pretty much every single race. It looks like it's won every single race and further. So you get not only kind of a degree of speed, exertion, but also of total energy spent in terms of distance. And then let me just show you one kind of fly race just so you can kind of get the gist of it. But yeah, you can see berberine and biotin here has won 11 of the races. Make that 12. Cool. Anything that you guys wanted to mention in terms of the setup of the races or how they work? Victor or Sam, either of you, feel free. I know I went over that pretty quickly, but anything you guys wanted to add?
Yeah, absolutely. If I may add, this is this is fascinating. The idea is we are measuring the speed as well as we see the health-wise also at the same time, right? So in terms of how these compounds are affecting throughout the lifespan, right? So this is not only one time point; it's throughout the lifespan, and then we follow this how it how long each of this compound can hold their effect in terms of not only the racing speed and the velocity, maybe the total lifespan as well, right? So that is the plan in the future too, right?
Yep. So the races, and we'll kind of get into this, but the idea with the races is to measure health on a continuous basis throughout the life of the animals. And with that, hopefully get a good predictor of what will actually help them live longer, right? It's very difficult to increase lifespan without increasing health span, meaning it's hard to live longer if you're not healthier. And so this is our way of kind of measuring health throughout the life of the animals to predict whether or not they'll live longer. And of course, the whole game here is to live longer. So this is kind of our way of doing it. Victor, anything you wanted to add on the setup?
I think all has been said.
Amazing.
That's just the setup we use. Probably just important to mention that all the tracking is done automatically with deep learning based algorithms. So we rely on that to do the measurements and then to get the insights of what's the health of the animals.
Amazing. So maybe in case people didn't catch that, all we do is take the videos of the flies. Victor has built algorithms that take the videos and can measure the path of each fly to get its speed and distance traveled. So incredible work. And this is kind of the result of that. So anyway, that's kind of an overview of our UI. As the experiments start streaming to the site live, there will be notifications that are sent through in Telegram and on Twitter. that will allow you to make sure that you're prepared and like on the site right before the new races drop. So that's coming soon. But what we wanted to do here first was go through all of the data that we've generated, basically the results from all the fly races. And basically compare which of the compounds are working better than others, and what are we seeing from early signals. All right, so here's kind of where we are. For this new batch of compounds that, most of which launched on whatever it was, July 31st, we've run 216 fly races, which is quite a few. The setup, kind of as we described, is you tap the tube of flies that you saw to the bottom. All the flies kind of like, basically aggregate at the bottom of this tube, and then they get 15 seconds to run around as much as they possibly can. We measure their speed and their distance, and yeah, basically they either get drug or control, and our goal is to find the drugs that make them move faster and farther. And yeah, just kind of like, why are we doing this? Why are we measuring the speed? Why does it matter? The idea is if the flies are faster and can move farther at each time point throughout their lifespan compared to control when they're given a drug, then that drug may be improving their health and may extend their lifespan. That's the point. And so here are all the different compounds that have been tested. So we've got thymoquinone, astaxanthin, resveratrol and berberine, cannabidiol, astrgaloside 4, berberine and biotin, berberine and punicalagin, silibinin, melatonin, rapamycin, ginsenoside RH2, and omeprazole. There's lots more drugs that are being tested on the flies. And, the Juvion team can tell you when those fly races will drop. But we're working on that. All of that's being set up at the moment. But this is the data we have so far. So this is by no means all of the tests we're currently running, but this is what we have data on so far. So we're going to go over this batch first. Cool. So just as a reminder, the current king of the pill for flies is urolithin A. Urolithin A has extended the lifespan of the flies by 20%. And so that is the benchmark to beat. If any compound can extend the lifespan of the flies more than 20%, it will become king of the pill, and it will get all the airdrops associated with king of the pill that urolithin A is getting right now. Okay. Cool. So the first token, let's go through biotin and berberine. I don't know if we have the dev on stream right now. Jillian, do we?
Let me check. I don't think so.
Cool. Anyway, we can get right into it. So in terms of the token, right now it's sitting at a 12K market cap, 114 holders, volume's fairly low. How did it do on the worms? It, fairly insignificantly decreased the lifespan by 5% on the C. elegans, and it's way too early to tell in terms of the survival. Only, over 90% of the flies are still alive in both groups, so can't comment on the survival yet. But in terms of the race data, what we're seeing is super interesting. So berberine and biotin has won every single race, meaning every time that the flies have been raced, it has traveled further and faster, meaning that the flies who are getting the drug being biotin and berberine, have gone faster and farther, and by a pretty significant margin. So 19% on distance and 17% on speed. And by the way, for each of these, we'll have the dose on the bottom right-hand corner if you care. So yeah, honestly, I was pretty surprised by this. I don't know if other people were as well. I know we should probably go through all of them to have some ability to compare between them, but this is a pretty interesting signal. After 12 races, already a very significant result. And by the way, if anyone has comments or has takes on the data, by all means, jump right in. No need to raise your hand or whatever. But yeah, huge congrats to Carlin, who is the dev for this. This is really, what we say is picking the right dose, let alone the right compounds to test, is shooting at a target in the dark. And when you hit a bullseye and actually get a good result, it's pretty impressive.
And Carlin is the part of the Raptor dev team also.
Exactly.
Which are doing a great job.
Yep, they're crushing.
Yeah, what I think is interesting, and I know you covered it, was this actually, when we looked at lifespan, right? This decreased the lifespan of worms if I remember correctly. Was that right, Benji?
Yeah, that's right. By 5%.
And so
not much.
I remember like being on that stream with Carlin, and it almost felt like it was kind of like, oh, but it goes to show you, I know it's still too early to tell and still too early to make any conclusions, but there really is a reason to push these compounds through the various animal models, because what happens in one, you might see a totally different result in another. So I would have loved to have had Carlin on this stream, because I do remember like all the hype leading up to that moment. And then it felt like, oh, but then, here we are. Like this compound's still very much in it. So excited to see this one continue to play out.
Yeah, totally. And as you move from organism to organism, what may work on one is not necessarily going to work in another. So yeah, I mean, like to your point, it's super important to keep testing. And yeah, I mean, a blessing that this had enough funding for the fly experiments also. Because we got this really cool result.
I think also it is good to point out that dosing may also impact here. So it could be that they choose the wrong dose here or something like that. But yeah, many different parameters why something may not work or not. So good to point out.
Yeah, especially when you get into combos, right? Like now you have two different doses that you have to pick, right? And so, but anyway, this seemed like, the right choice. Without any others that were tested. So yeah, huge congrats to Carlin. Data looks great. Doesn't get cleaner than that. Cool. The next one was berberine and punicalagin. I don't know if anyone has insight into the chemistry or like the origin of either of these. Definitely didn't do my own research there. But yeah, similar story here where the data didn't look great in the worms. Market cap's at about 11K also. And there's only been about 3 races. So definitely too early to start drawing any sort of conclusions, but so far pretty good. Next is thymoquinone. So this comes from like black seed oil, as far as I know. And yeah, market cap's a little bit higher, 15K. Had really no effect on the worms and still too early to tell on the flies. But what's been interesting for me is that it doesn't really seem to be having a positive effect on the flies. At least at this dose. But yeah, definitely like decreasing in terms of speed, non-significant on distance. But yeah, I found this pretty surprising just because, I mean, if you go on Twitter, you'll see tons of people raving about thymoquinone, especially for like dermatological conditions. So yeah, this was definitely surprising to me. But anyone have thoughts on this compound?
Yeah, and I know this dev isn't on the stream, but they're out there watching. So if you have any comments too, we can be sure and show them on the stream.
Yeah. Anyway, she's been doing a great job. Like definitely building a ton of hype for this compound. And maybe we didn't test the right dose. Maybe this isn't the right experiment to showcase its true power. But yeah, definitely not the result I was hoping for. Cool. Astaxanthin, definitely like a big compound in terms of like longevity excitement. And so I don't even know who this dev is. This was definitely one of those anon devs who slid into our Twitter DMs and was like, oh, can we launch? Probably looked up on ChatGPT what they should launch. Anyway, here we are. And haven't seen the dev since. So anyway,
hey, anyone with an idea.
Yeah, you get a variety of people in the DeSci world. So yeah, the market cap here is pretty low. It's at 6K, probably because the dev hasn't been doing any sort of marketing. And yeah, we are testing this on the worms at Aura right now, and still too early to tell on the flies in terms of lifespan stuff. But the trend looks decent. We'll have to see. It's probably too early. There's no strong signal yet for astaxanthin on the races. Next is Troll. Troll definitely cruising at a much higher market cap. Reached a pretty high peak, but definitely still like at a much higher cruising altitude than the rest. What's really impressive is the number of holders. So like I think most of the other compounds have between 100 and 150 holders. The dev here, Raheem, has really put in a ton of effort and has been getting great results on community building. And yeah, has built this to almost I don't know if he's broken 500 holders yet at any point, but yeah, this is a pretty solid community behind resveratrol and berberine, so props to him. Not a ton of signal that we can tell yet on the flies. It's way too early. And on the worms, yeah, we didn't see much, but the data is looking pretty strong on the races. So, 12% increase on distance so far, significant result already, only after 15 races. And same thing for speed. So super exciting for Raheem. I don't know if he's on the stream, but good
is.
work if he's listening.
Let's go.
Let's go. What up, Raheem? Congrats,
What's up,
man.
guys? No, this
Dude.
looks good. Thank you.
No,
No,
good to
exciting
have you.
first results.
Yeah, no, props to you. Pick the right dose. You hit the bullseye in the dark. So congrats, man.
And you are
This is it.
consolidating this token to Genesis Labs, or what's going on there, Raheem?
Yeah, so we will be continuing to so Troll will be continuing to be supported via Genesis Labs. So as we progress from flies into mice and further experiments, yeah, it's going to be one of the projects that's supported. So yeah, interesting to see that we're getting closer. We're getting closer with the dose. Results are improving. So interested to see how it continues to perform.
More holders to that Genesis Labs then. Great. Cool.
Exactly, yeah.
And one comment here. So the thing until now, we saw three combination, including that include berberine, that show positive effects in the speed, in the flies and the racing. To me, I think that actually berberine is one of my favorite longevity compounds. I truly believe that this work in mice and probably in humans as well. I wonder why nobody launched berberine alone, because to me, I think that this experiment should be done, because and then to see if we can massify the effect combining with another thing, like for instance with this resveratrol.
Yeah, I think the idea is like, can you develop any sort of IP from the combos? So I think that was the objective, is to like find the right combo and develop IP around it. But yeah, no, I totally agree with you. They call berberine nature's Ozempic, or at least that's what's trending on X. So we'll see about that.
Awesome. Berberine to me is very similar, like metformin as well. So the mechanism of action. So no, it's very, very interesting. I would like to see in parallel racing the three combos of berberine, comparing the flies. This could be fun.
That would be fun. That would be fun. Cool. Anyone else? All right, let's go. Cool. CBD, another Anon dev who just showed up and was like, let's launch CBD, and launched it, and haven't really heard from him again. And it's probably why his token's at a 4K market cap. But yeah, only 50 holders here. It's actually being tested at Aura as we speak. And yeah, the fly race just started, or excuse me, the fly experiment just started. So way too early to get any sort of lifespan data. And yeah, probably still too early for the fly race as well. But if there's any trend, it's probably looking like you should stay off the weed. So good to know. Good to know. Good to know. Any thoughts, Jillian, on staying off the weed?
No, I was just gonna say. I mean, I know it's still early. It's only three fly races in, but it's it's in line with what I would expect, which is why stay
this
off
isn't
the
a
weed.
surprise. Yeah,
No,
if I was running
I
a
would have been
race,
surprised. Yeah, I was like, I'd have been surprised if it made them go faster and like go crazy. But I was like, well, yeah, this is about what I would expect.
Yeah, I don't know. If
We'll
I
see,
was
though.
if I was racing F1, I probably wouldn't want to have some weed beforehand. Cool. Any other comments?
Just brought up a couple other folks too. We have Christian here and Victor. I know you might have been in, but it looks like you weren't on the stage. So excited to bring you up.
What, Christian? What timing? What timing? We're ready for you, sir. Very nice to see you. Thanks so much for joining. Yeah, so basically astragaloside 4 comes from the astragalus root. Maybe you want to talk a little bit about like where the molecule comes from, what like the inspiration was behind launching it?
Yeah, thanks for the invitation, Benji. I just came from another presentation, so sorry for just jumping in here. So it's actually a natural product, and as a former natural product chemist, I was always intrigued by traditional Chinese herbs, and it's actually coming from the dried root of a plant that has been used in Chinese medicine for over 2,000 years and has different dry terpenoid saponins, and astragaloside 4 is one of the components that has shown most pharmacological activity across many studies, actually. And I still haven't managed to make a nice presentation on that, but I'm planning to do that because it has been really studied in many papers, both in vitro and in vivo. Of course, you always need high dosages, because also absorption is not ideal, and for natural products, it's not very difficult, not very easy to to reach exposure levels that you would need for a standard small molecule drug. But I think there's still a lot of value to explore this a bit further. And now I'm very, very much looking forward to the first results. Yeah.
Awesome. Great. So thanks for sharing all that. What's interesting is I'm not sure how much it had an effect on the lifespan extension. Maybe there was some early signal. And by the way, what this graph is at the bottom is the fly's survival mean life extension percentage. So we just take whatever the mean lifespan is of the animals that are being tested so far, and we just plot it just that it's a single line rather than a Kaplan-Meier curve where there are two lines. And yeah, it doesn't seem like there's a ton of signal, maybe early, but not as it got into like max lifespan territory. But what is interesting is it definitely made the flies faster and move farther. So both results were pretty significant. I think you probably saw this, Christian, from the data. But yeah, here it is kind of like at a statistical level, like definitely significant. Increased, both the speed and the distance significantly. So congrats on that result. Super interesting. Seems like you nailed the right dose.
Super. Looking forward to the mouse studies. Yeah.
Yeah, same here. Astragaloside 4 has received enough funds for the mouse experiments, so we'll be running those shortly as well.
Do we have an update on when those would start? You got a when girl here. Sorry, guys.
You know who to ask.
You mean for the mice?
Yeah.
Yes, sir.
Yeah. No, the reality is that we received an animal of 12 months a couple of weeks before ago. Now we are in this period that we don't want to touch because after the transport and this is a new animal facility for them, we don't want to bother them. And maybe next week we are going to start to measure body weight, body temperature, to have a baseline and to start the treatment in the moment that we have all this data. But yeah, so the experiment will start soon.
Let's go. That's amazing.
Let's go.
I just had a visual of like, when fighters go for their like weigh-in, like when they're getting on the scales and getting their like little baselines.
Yes.
Paris, maybe we could show the community like the mice weigh-in.
Yeah.
Is it possible?
We can.
Those are always feisty in UFC. So
yeah, seriously.
Maybe we can rile up the mice, get them all feisty.
Real quick, we did have a question from the audience here. Christian, do you want to take this one?
Christian,
so this is I can see the question. I can reply. So always that we talk about natural compounds and this, the people think that they are safe or because they are natural, and the reality is like they are not. So they think that there are natural compounds that could be safe, or they are not. There are another one that they are not. Depend on the concentration. The good thing of this longevity study and this performance when we do it in mice is like we are going to analyze these kind of things, because maybe even they cannot be toxic for liver in this question in short term when we do only one treatment, but in chronic effect maybe we will have something. But in the mice, we are going to analyze in the plasma this effect. If we start to see some parameters that cause liver problem, we are going to observe and we are going to see. So we are going to reply this question with the mouse experiment.
Amazing. And we can't answer this one with the flies, right, Sam? Like, do flies have livers?
Flies do not have liver-like organs, but they have the fat bodies, and it's called anocytes. So flies do not have bloods; they have hemolymph, right? So, but it actually does the same thing. A lot of genes and proteins been discovered associated with the liver function been discovered in flies, but the structure is different. It's called differently. It's fat bodies in the dorsal region of the body and anocytes sitting there.
Cool. Is it like the equivalent of like the biliary system, like the bile?
Yeah, equivalent of that.
Got it. Super cool.
I mean, to that question, if I may jump in, one piece of information I would like to share actually, there was a study by a Chinese group using astragaloside 4, I think at the 50 mg per kg range in a liver fibrosis model, and there they actually saw reduced collagen deposition and some of the cytokines were downregulated. So there could be some potential for antifibrotic, anti-inflammatory effects, but of course, yeah, we will need to see what comes out of the mouse studies.
Totally. But your hypothesis is that it's actually good for the liver, not that it's not bad.
Yeah. Exactly. I think it's should be good for the liver.
Amazing. Cool. So we invited the scientist behind melatonin, but unfortunately he fell asleep. So I think that's actually bullish for the molecule. So yeah, this is melatonin. It's cruising at about an 8K market cap, decent amount of holders, 150. We saw some pretty interesting signal with the worms with melatonin. It's of course like a body hormone, an endogenous hormone, and known antioxidant. So I think the theory behind like the scientist had was that it would increase lifespan because of its potent antioxidant activity. And definitely got some strong signal in the worms. Too early to tell in the flies. Trend is looking good though. But what is interesting, and I would not have guessed this, just knowing how melatonin works, but seems to be making the flies faster. So I don't know if they're sleeping better, or oxidative stress is like more of a thing in flies, or if they've developed tolerance to it and now they're just powering through the melatonin tiredness. Who knows? Any guesses from the aging scientists in the house?
So no, to me, melatonin finally organize the cycles of the night and light and make the animal and us to sleep better. And we know that good sleep is one of the most interesting anti-aging therapy. So I'm not surprised that melatonin does something in the flies, because
Totally. It's actually, you make a good point though about circadian rhythms. I know flies have like a very interesting circadian rhythm where they're most awake during dawn and dusk. It's like a unique kind of circadian rhythm, or like more rare. But I guess, when do the flies get tested, like time of day? Is it during their active period or is it during their inactive period?
Victor is a behavioral scientist, so he will answer
Yes,
this.
so they are tested right after the peak of dusk. So it's basically they are still active, so we just make sure that they will behave at the best performance basically that they will have. But it's not on the very, very peak, because that will be very noisy. So it's a little bit after that.
Gotcha. Super interesting. Super interesting. Well, melatonin seems to be making the flies more healthy, whichever way you look at it. Cool. Speaking of the liver, this compound, also Dev has completely ghosted. If you're the Silibin Dev and you want to get more involved, like, please hit us up. But this is from milk thistle. So a lot of people take milk thistle whenever they have like a strained liver or are taking drugs that have known liver toxicity, because this is supposed to milk thistle, silibin are supposed to promote liver health somehow. I actually forget the exact mechanism. But yeah, this compound's trading at about a 4K market cap, not very many holders. despite having some pretty good data in the worms. And I mean, only 3 races, but pretty strong effect size so far in the flies, which is interesting. So probably shouldn't comment too much. Only 3 races, but I mean, man, that was pretty good so far.
Run it back.
If you are the Libyan dev, please hit us up.
Yes, would love to see this one come alive.
Yeah, the
I mean,
data's
it's
good.
clearly
this is a good choice. Yeah.
That's what I think is so interesting is like, based on how obviously like the protocol is designed, if there's enough fees, the science will continue. So
the show goes on.
The show goes on with or without you. And yeah, we're doing real science that's like pushing information and a body of evidence forward. So you might come back around and be surprised, folks.
Yep. Cool. All right. Next, we've got ginsenoside RH2. This comes from ginseng root. The market cap's definitely higher than the previous one at 11K. Had unbelievable data in the worms. This was definitely surprising. I mean, Christian, you might like this one. Comes from, Eastern medicine, natural product. Has been used for, many, many centuries as like a known therapeutic, like kind of homeopathic therapeutic. The data was incredible in the worms, and in the flies, it's looking good too. Probably too early to tell.
The dev team is here.
Oh, sweet.
Yeah,
Ashish
looks good. Yeah, I'm here.
Oh, what's up, Ashish? How are you?
Good.
Yeah.
It's quite interesting to see. I mean, this compound is coming from like red ginseng, one of the known molecule or compounds from like centuries, which has effect on longevity, because like ginseng is very well known in like Chinese or Asian medicines for longevity effects. And science-wise also, like it has a lot of known like pathways, like it inhibits the mTOR pathway, same as rapamycin, and a lot of protective effect it has on our cell, like anti-inflammatory, immune modulation, and so on. So good to see like it is moving ahead in the flies and looking forward.
Awesome. Cool. And wait, Jillian, I saw a question from the comments that looked good, and it's from Zach. It says, how you gonna make those flying higher though? I think what he's asking is like, how do we make the flies run? Yeah. So I don't know if like what he meant by high, whether it was when we were talking about CBD, or whether he was just asking like, how do the flies actually know to move? But I realized we should probably address that. Victor, Sam, you guys want to talk about like what actually is the stimulus that makes the flies run?
I think he meant how healthy the fly is. They have this natural tendency to go like to the upside, to the mouth of the capillary. So how the fitter the flies are, they perform like better.
Totally.
And Benji, you might have covered this at the top of the call, but I think even talking through like the geotaxy study itself, like how the device works to capture the flies, knock them to the bottom of the tube, and then cause them to run up. I think maybe walking through that would be good. Or Sam or Victor, if you guys want to explain a bit about that too. Look at this beautiful graphic.
Sam, are you on mute, boss?
All right. Victor would have been a better person to answer this, but the flies have this innate behavior response to go towards the light, and that's the geotaxis experiment, right? So that is one of the things, you can tell when you see the behavioral data on flies. You can't say the flies are not moving because they are lazy or not. Everyone has to move, and that's their innate endogenous behavior. So that's why this geotaxis is really crude way of measuring the health of the animals. I hope. Yeah.
Yeah, it makes a ton of sense. It's just instinctual.
Yeah.
So like they have this instinct and they want to move. It's just a question of whether they can.
Yeah.
Yeah. Awesome. Cool. So yeah, back to results. Ginsenoside RH2 looking pretty good. Definitely a positive signal, about 6-7% on each. Definitely need more data to get it to see if it's significant, but trends looking amazing. And then rapamycin. And rapamycin, definitely a higher market cap than a lot of the rest, 50K, almost 300 holders. The data in worms was incredible. It was king of the pill for a bit until dethroned by omeprazole. But so increased by 35% the lifespan of the C. elegans. And the trend was not looking great for the flies at the beginning. But it's starting to trend in the right direction now. We're at about, I think, 5% mean life extension at the moment. But this is for this one dose tested. So rapamycin was so important that we tested it at multiple doses in the flies. And so we'll be getting the data on the other doses soon. But yeah, I mean, like it's we're expecting big things of rapamycin when it comes to the lifespan extension.
And the rapamycin longevity lab is also part of that dev team. And if we look, for example, at the worm data, worm data when it came out, that is the best medium lifespan which ever has been recorded on that compound. So yeah, we did a historical moment there when the data came out. And,
yeah, that was a really cool moment.
Yeah, and yeah, I think the fly data, it showed around 15% or something like that in median lifespan. When I did a quick calculation there, but yeah, something. But we'll test some different doses and also one other strain to see how things translate. So yeah.
Yeah, awesome. So yeah, maybe just to recap what Christopher said, not only are we testing rapamycin in multiple doses, but we're testing in multiple strains of fly. We're just trying to see, what does the signal look like? Does the signal that we saw in worms and people are, have seen in mice, does it hold in flies as well? But the really interesting thing about the races is that the signal is super strong there too. And so I think this is the strongest signal we've seen of any of the compounds so far in the fly races, where the speeds increased by 38%. The distance traveled has increased by 21%. So yeah, I mean, like rapamycin seems to be maintaining health for longer in the flies, as measured by, these negative geotaxia assays to a very significant degree. I mean, look at the p-values. They're like super significant. And, almost 50 races tested thus far. So it's looking pretty good. Sam and Victor, is this surprising to you? Did you expect this? Have you seen this before? Give us the alpha.
Yeah, rapamycin is one of the sort of holy grail of longevity, right? So I think it is, we have tested that so many times, and this is absolutely, we have observed over and over. I think as Christopher was saying, the different concentration and different strains of and testing this rapamycin will be very, very important, because this is something, we have seen it over and over. And it's very exciting. Yeah, I think nothing to add anymore. I think so many literatures, so many, papers came out based on this. And I have tested this compound so many times. So, I mean, it's obvious what is it.
We're beating a dead fly, you can say.
Horse fly.
There is a reason why I call the lab rapamycin longevity lab.
Yeah, it's the gold standard.
Yeah.
If I could add something to that, guys. Sorry, it's Garish. I think this is super intriguing because I just wanted to highlight at AgelessRx, we ran a 12-month clinical trial in humans, obviously. And we saw improvements in muscle mass in postmenopausal women. So this is a time period when muscle mass is typically, in accelerated decline. And we didn't just see a maintenance, we saw an improvement within all women within, the 10 milligram compounded rapamycin group. So super interesting because it seemed to have a dose specific effect within that trial. It seemed to improve muscle mass where there's been some pushback on how rapamycin might affect muscle, might affect exercise-induced benefits. So, this data showing improvements in distance and speed in flies kind of merges with some early results we've seen in humans as well, suggesting performance benefits amongst the many, many other things we're seeing. So I think this is a cool opportunity to just get some more attention on these interesting effects, these dose-specific effects. And excited to see this kind of converge with some of the data we've collected as well.
Yeah.
Anyway, I think that's super. Sorry, go ahead, Benji.
All I was going to say is this was surprising to me because when you think about what rapamycin does molecularly, it activates mTOR, right? mTOR causes catabolism, basically eating your body. And so my thinking was, okay, maybe rapamycin extends life, but it'll probably not increase your health span as much, right? It will, eat at all of your tissue, including your muscle, which my thinking was, that would probably, make the animals run about the same speed and distance, but maybe live longer because, they're metabolizing slower. Also the whole metabolism piece, right? If metabolism's slowing down, how are they moving faster and farther? For me, this was counterintuitive, but
I think
your rationalization.
I think here the key thing is the dosing. If you dose rapamycin too high, then you will get the detrimental effect. But if you find a sweet spot, then I think you can get the nice health span and lifespan effect.
And just to add to that, I think something that's really important is with exercise, with recovery, with performance. I mean, when we're exercising or when we're pushing ourselves, we're creating little tears and damages. And then it's that recovery period when there's actually a boost in autophagy. There's inhibition of mTOR signaling. There's recycling of the damage that's done to produce, newer, better, higher quality components. So that's what this optimized dosing of rapamycin may be able to do is just help with that regeneration and recovery. So yeah, super interesting to see.
We got this question from someone in the community. Why is Brian Johnson downplaying rapamycin? And I wonder if it's some of what you guys have talked about would be curious to hear your guys' thoughts.
I would like to jump to this question as well, because always the people say that there are good drug and bad drug. And the reality, this is not like that. There are good dose and bad dose. So the thing that rapamycin is we discussed before, can have a very good effect at one dose, but then if we use chronic and high dose, maybe there are some side effects that can be more negative than the positive effect of rapamycin. So, and believe me, nobody did this experiment before in humans or in animals to do chronic effects in many different doses. So maybe we are going to be one of the first to verify if this could be good or bad.
Yeah, I think also one thing which, when it comes to Brian Johnson, I have written a quite good X post around this, which I can post later on here. But the thing here is that he takes lots of different other longevity interventions that also inhibit mTOR. And as Paras said here, if you start to hit mTOR from lots of different places, then you can get too much mTOR inhibition and get the detrimental effects. And he saw some type of side effects like
cardio, I think it was.
Cardio, maybe, but I think it was glucose levels and lipids levels went a little bit up. And those things may be indications that mTOR inhibition is too high. So, yeah.
Totally. The dose, the poison is in the dose, I think, to both of your guys' points. I think that the only piece of pushback I would have the dose being the issue is Brian said that he tried many different doses. He tried the pulsing, which is like where you take it once a week. I think he was trying like six milligrams or something, or 6 grams. I forget what the order of magnitude is. And then he was also trying the daily doses as well. And he said he saw, elevated heart rate, I think was his biggest issue. But yeah, I mean, to your point, it could be because he's taking other things like berberine that we also know inhibit mTOR2, that just, basically multiply the effect to the point where it crosses, being good for you and promoting health. So
this is I have a question for Kriste. So because rapamycin is taken for humans for to immunosuppressor in transplant, in human with transplant. So there are reports of side effects like Brian Johnson reported, or maybe it's like you say, that Brian Johnson is inhibiting the mTOR for many different places, and because of that, it's a very high dose.
When it comes to chronic use of rapamycin and high doses, which organ transplant patient use, then you can see those two side effects. Glucose levels goes up and lipids goes up, and also that you can get mouth sores and things like that. So there are different things you can look at, but then I think also one thing Girish points out in different times is that personalized medicine is also important, because some people may not need rapamycin because they don't have or they use other interventions which works good for them. Let's say that they are very healthy persons, and by that you don't need rapamycin.
Totally. If you're already doing caloric restriction.
Yeah, also if like in the human data, it was previously mentioned that it kind of sort of increased the muscle mass. It would be interesting to see the muscle strength with rapamycin too. Does it have an impact on that? And second, the rapalogs, right? So there are so many rapalogs have been really, really important to see how they're affecting in terms of the same two pathways.
Yeah. I think there exists some mice data around muscle that the mice is not losing muscle on rapamycin. But again, here we come to the dosing.
Yeah.
So yeah.
And just to add to that, we have at Ageless RX that is over 1,000 patients taking rapamycin, and we have various degrees of rigorous data that we're collecting, like body composition and DEXA scans are some of the more rigorous variables. But then we also deploy these quality of life assessments as a patient is on rapamycin, and we see improvements, self-reported improvements in like recovery and energy and pain and all these aspects of quality of life that, a lot of the times get underplayed in the longevity field, but are so crucial, right, to like overall health span as you age. So these are definitely some of the things that are plugging in here as well. And again, we're seeing people on the same dose of rapamycin having different blood levels of rapamycin, so the bioavailability differences. So there's so many layers of complexity here. And as Krista mentioned before, finding out these personalized dosing strategies are super, super important. And I'm encouraged that there's more clinical trials that, like Brad Stanfield, Sam, to your point, he's going to be looking at strength in elderly individuals. I'm encouraged that there's more clinical trials looking at this, but also we need to be digging more into the preclinical studies and seeing where rapamycin may be effective, how we can optimize dose, and that'll give some pretty good insights as we continue making a case to run more human studies and collect more real-world data.
Amazing. Thank you, guys.
And Benji, just to come back around on this, so there's 3 fly studies for rapamycin. Is that right? Underway?
I gotta go back and look how many there are. I think it might be 2 doses, 2 strains. So I think 4.
4. Okay. And then, just a reminder, I know you covered it at the top of the call, but the current king of the pill is urolithin A. and the median life extension to beat was in flies was yeah, you got it
20%.
there. 20%.
I guess, Sam, question for you is, have you observed more than a 20% lifespan extension in the flies before with anything? You don't have to say what it is, but have you seen it higher? Also, you're on mute, boss.
It depends what you're talking about, the median lifespan or total lifespan.
Median lifespan.
In my experience, yes, we have seen pretty much like more than one time. 20% is a solid benchmark, but I think with the right combo and right concentration, we can reach to 40 to 47%. That's the highest.
Oh, wow.
For the median lifespan I'm talking about.
Well, if you ever wanted to submit a compound to Pump Science and find out for sure, you know where to find us.
I mean, it would be, I'm like waiting for the day that Sam says that the phdgens found something that beat what he's done so far. So we'll get there. We're going to get there.
We'll get there, but it doesn't look like we're going to get there with this dose of omeprazole, which is definitely not extending the lifespan of the flies. So yeah, just kind of as a reminder, omepal is the token for omeprazole. The market cap's just over 100K. Definitely one of the most prominent communities within pump science. And yeah, it's the current king of the pill of the worms. Definitely the highest recorded that we've ever seen in terms of lifespan extension there. But omeprazole, like rapamycin, we've tested multiple different doses. We're trying to find the right one. Want to see the one that has the best effect, and it looks like we may have overshot it here. So we're all talking about how important dosing is with mTOR and inhibitors, and we may have overdone it a bit here. So anyway,
no, but the thing we did here was to. Yeah, our strategy was to test high dose and low doses. So here, we tested the high dose, not in parallel with the low doses. But in the future, we will do high and low doses, in parallel. So, definitely. I think we have, I think it's 6 different experiments in flies that we have in the pipeline. So, lots of interesting data will come out, and those doses will most likely result in some type of lifespan extension, because we have some unpublished data in the background. So
amazing. So you know better than any of us. We'll have to take your word for it, but won't believe it till we see it. Awesome. Well, yeah, I mean, even despite the suboptimal lifespan data that we have so far, the racing data is looking pretty solid. Not quite significant yet, but definitely interesting from a trend perspective.
But this is very interesting in the sense, because as you see, what happened to omeprazole, the flies just do not start dying early, and suddenly they fall, right? So it drops suddenly. So basically, it's definitely a dose-dependent problem, right? So that's why it is very, we are doing different concentration so that it can hold the behavior for a long period of time, and we not see the drop of this fly lifespan suddenly.
See if we can see what you're talking about.
It's an interesting point, Sam. I think, that's like a classic sign of toxicity that we've seen in C. elegans intervention testing program as well. You get this early drop, and then interestingly enough, the flies that are surviving are showing, some trends of enhanced health. But hey, this is the good thing, right? I think defining the optimal dose window is really important, because say we stopped at 25 micromolars of omeprazole, and that's not the optimal dose for longevity. This is a good way to test the upper limit and the ceiling. And then, we have the 5 micromolar as well for the floor. And then we'll find some place in between that's the optimal zone. So all in all, it's good we have this data. I mean, of course, we run it in parallel and release it in parallel to the public, but I think it's useful information nonetheless.
Oh. My computer's spazzing. anyway, wanted to check out that Kaplan-Meier curve that you guys were talking about. So is what you guys saying, because this starts dropping so early, this means that there's kind of like a toxic effect here? This isn't something that you would expect to see if the dose is right.
You're muted, Sam.
Oh yeah, so I just explained once. Yeah, I think this is the toxicity in the early stage, and then you see the drop really significantly from day 16 or 20 onwards, if I say it correctly, and then it holds, and then it just dies off, right? So you don't see that in controls at the time point.
Gotcha.
I think that's why the behavior should be. We're probably going to see. I don't know if we have that yet. I mean, the behavior is going to be really, really getting worse and worse. Towards the end of the lifespan. Hopefully.
Let's see.
If it is not, I'm just going to log off now.
Well, it looks like, at least for the flies that are alive, they're keeping pace. But yeah, no, I mean, you're right. I mean, with both of these latest few, it's not looking good. We OD'd on me, pal, on the flies. Good to know.
But anyways, we're going to have many concentrations and different concentration, different strains. We'll be able to totally extrapolate what would be the best combo in terms of moving forward.
Awesome. So wanted to show what we have as kind of like the rankings just based on the races. This is not based on the lifespan data, but just the races themselves in terms of which are the top performing compounds for the data that we have. Top in both distance and speed is rapamycin. Berberine and biotin is number 2 on distance and number 3 on speed. Silibinin is 3 on distance and 4 on speed. Berberine and punicalagin is 2 on speed and what is that? 1, 2, 3, 4, 5, 6 on distance. So yeah, we'll basically keep updating these plots over time and posting them. But you can think of these as like power rankings of us just ranking top to bottom performance for each of the compounds. And when you sum it all up, here are the top fly standings thus far, kind of in order, if you combine speed and distance. But rapamycin, berberine and biotin, silibinin, berberine and punicalagin, and astragaloside 4. So these are the top 5 performing thus far in the fly races. So with that, wanted to make sure that like none of what has been said is financial advice. Definitely do your own research. This is the summary of ours, and wanted to give a huge shout out to Juvion Health Sciences for putting on this incredible opportunity for us to be able to run these experiments, stream them to the people, and yeah, hope they're finding them interesting. But yeah, thank you guys so much.
Yeah, it's super great work with fly experiments. So.
I would like to say something though. That I was doing science and experiment for over 20 years already, and it's the first time that I see a very high quality scientific data and make it attractive for the audience. If someone is not here scientist, that come on, this is good. And I think that this is the way that we have to go, that we are here people discussing about the result and even trying to improve to what is the next experiment that we can do or how we can do it to do it better. Congrats, Benji.
Congrats to all of you guys. Couldn't do this without all of you. So yeah, but agree with you completely. Like, it's super important to make all these results super interpretable. I mean, you should see what the data looks like coming out of Juvion. It's how we asked for it, but it's just like spreadsheets and spreadsheets and spreadsheets. But we think that this is like the race interface, and kind of like the plots here are a better way to bring other people in and actually make the good results attractive, but always open to feedback. And if there's any other way we can improve it or present it, we're all for it. But yeah, the goal is to gamify this and make it feel like a video game.
The game is don't die.
The game is don't die. It always has been.
Real quick, we did have a comment come in from the Maquinone drug dev. So I'm going to pull this up here. I don't think they were able to join, but it says, sorry for the late comment. I think we're still early in the TQ fly run. No significant early toxicity detected. And I'm optimistic the second half of the fly experiment is where it shows, if it shows up. There's very little direct longevity research on this compound. So this might be one of the first clean lifespan fly reads. Small side note, prior to ingestion, light can degrade the maquinone on the food. If this dose doesn't cut it, we may have to increase the dose. So I'm not sure if anybody has any comments on this, but thank you so much for sharing. We see you out there.
Awesome.
Yeah, very good point about the light sensitivity. Absolutely, we're aware of that.
Are the flies kept in the dark?
No, flies not kept in the dark, but when you start, developing the fly food with the thymoquinine, we took the proper precaution, wrapping it up, make sure you're not, put it outside so that it gets deteriorated. So we're aware of that. You can't put the flies in the dark because of the circadian clock, then it's totally gone, right? So you got to maintain them in a 12-hour light and dark cycle. We cannot do that with thymoquinine, but yeah, it was a good point.
Awesome. Yeah, no, we'll tell her if she can get the funding for it to submit a higher dose. If it's true, and yeah, there isn't really a ton of toxicity seen, like maybe we should just up the dose. So we'll let her know that. But Sam, Victor, any final thoughts from you guys in general? Like, can be specific, general, before we sign off here.
No, I think it's fantastic, as Paras put it in such a way that it's such a platform that we're not just testing, we're all of us putting our head down and try to understand the complexity of longevity science. We're trying to put it together in a very simple way and try to learn it from here to make it better and better. I think that is something is very, very interesting. That's all I could say. Yeah.
Awesome. Well, thanks for sharing,
I'm
guys.
smiling.
And thanks so much for joining.
Yeah.
I was going to say I'm smiling, Sam, because I had a moment just like you earlier this week where my son came into my office and I had the fly experiments on Pump Science up, and he walked in and was like, "What is that?" And I was like, "This is my job." And so I was like explaining to him like what Pump Science was, and he was like, "Can I see the race?" And we hit play, and it was so cool to see him be like, "Oh, this one's better than that one in this race." And then just kind of like see how easy it was for even like him to understand. Like obviously he was like, "What's going on in the middle? Are those real flies?" And yeah, I just had like literally, I'm having like deja vu because yesterday I had the same moment where the son came in, was on my lap, and thought the science that we were doing was so cool. And it was even cooler to explain to him like why I was doing it. And like the goal is, to really help us all live longer, but even leave his generation with data that they can, hopefully push the field even farther forward. So yeah, it's just like what we're doing. You guys said it really well, is, not only doing real science and sharing it, but making it in a way where it's accessible, digestible, easy to understand, and fun. Because at the end of it, if we can't keep people engaged and, watching what we're doing, then we're going on a journey alone. And it's really a lot more fun when we bring people with us. So huge hats off to everyone on this call. We wouldn't be having this call and doing the science if it weren't for you guys working with us, bringing the ideas, building the community, keeping people engaged. I'm just so grateful and in awe of all of you. So thank you so much.
Awesome. Go team. We'll see you guys for the next one. We'll have more data dropping soon. We'll do the same thing where we go through it again, show you kind of which ones are working, which ones are not. Hopefully we have some really exciting results to show. And so, yeah, stay tuned. And thanks for tuning in. We'll be back in probably a week or two.
Off to the races.
Sayonara.